Good morning, everybody. Nice to see you here for our departmental grand rounds. I'm just going to briefly mention that after our presentation today, after the QA session at around 9:30, there'll be a session for trainees across the hall with our grand round speaker. And with that, I'm going to introduce the trainee who normally introduces the grand round speaker. This one you've seen before, Dr. Murray Steen.
Can you hear me? Yeah, okay, great. So I'm really happy to be able to introduce our grand round speaker today, Dr. Jordan Smoller. Jordan has been in a rut for quite a while now. Let me sort of explain. So he did his undergraduate studies at Harvard University, went to medical school at Harvard Medical School, did his residency training in psychiatry at McLean Hospital, a Harvard-affiliated hospital. Obtained his doctorate in epidemiology at the Harvard School of Public Health. He's sort of seeing a pattern here. Poor guy can't get out of Harvard. He's moved up rapidly through the ranks over the years, and he's for a while now been professor of psychiatry and professor in epidemiology at the School of Public Health. Jordan holds the Massachusetts General Hospital Trustees Endowed Chair in Psychiatric Neuroscience. He is director of the PINGU, which is the little symbol there on the left, the Psychiatric and Neurodevelopmental Genetics Unit. He's also director of the Precision Medicine Unit, something he's going to talk to us about this morning at MGH in the Center for Genomic Medicine. Jordan has written extensively. He has over 350 scientific publications. He's also published for the popular reading crowd. He wrote a book called The Other Side of Normal that talks about interesting concepts related to personality and development and psychiatric disorders. He's also co-written with his mother, who herself is a distinguished epidemiologist, a textbook, a primer on biostatistics and epidemiology that's now in its 4th edition, and I highly recommend. I have a signed copy. Jordan also has numerous scientific leadership roles. I'm not even going to go through all of them, but he's currently vice president of the International Society of Psychiatric Genetics. He is co-chair of the All of Us Science Committee. All of Us is the big genomics biobank program that's happening in the United States with the aim of collecting probably over 1 million people. UCSD has one of the collection sites here. So Jordan has numerous scientific leadership roles beyond that. He's extremely well suited to being in those kinds of positions, given he has a sort of a very broad scientific knowledge, great depth of knowledge. He's extremely good at being able to explain very complex things to individuals. I've been a beneficiary of that many times, and I think many of us will this morning as well. But importantly, I think the reason he turns out to be in so many scientific leadership roles is that he really is a very genuine, honest, caring person, and he makes people want to work with him and work with each other for him. And I think that that's probably one of his greatest strengths. So please join me in welcoming our grand round speaker, Dr. Jordan Smoller.
Thank you, Murray. I think that was the nicest introduction I ever got. And you, as a trainee, you have a great career ahead of you. Well, it's really a pleasure to be here. And I spent a wonderful day here yesterday meeting with folks, many old friends and new faces as well. And it was really just a total pleasure. And I got to talk to my mentor who got me into psychiatric genetics, Ming Shuang, who couldn't be here today, but really influenced me greatly. And so it's just great to be back at UCSD because I've been here before. Despite what Murray said, this is the second time I've left Harvard, I think, in the last 30 years. So, but it's a great community and wonderful to see what everybody's doing here. So the title of my talk today is Precision Psychiatry, Is That a Thing? And the reason I titled it that is, I mean, I don't know if you're beginning to hear this term of precision psychiatry. Certainly precision medicine is something that we're increasingly hearing about. And I am one of the people who does think that precision psychiatry is becoming a thing. And I want to talk with you a little bit about that. So what I'm going to do is really kind of give you an overview of what that might mean, but focus on a few different stories or projects that we're working on to try to advance that concept or that field. So I'm going to start by saying something that you know well, but just to set the stage for why a slightly different approach to psychiatric illness is something we urgently need. And you know all this undoubtedly well, but the disorders that we treat are very common, lifetime prevalence of more than 50%. They start early in life, most of them. They're responsible for just enormous morbidity and mortality, incredible healthcare costs, not only death from suicide, but just death, premature death associated with severe mental illness of a decade or more, astoundingly. And as we know, most of the treatments that we have, with very few exceptions, are based on biological insights that are decades old. And we know that while they are very effective for many people and life-saving for some people, in each of the disorders, the story is certainly not where we would like it to be. So there's just an enormous need to begin to fill these gaps and to accelerate. Now, this has been going on for some time, and so it's not something brand new. There are other gaps that we don't really have traction on and need to. Tools for identifying risk of disorder and important psychiatric outcomes, finding genetic or other predictors of treatment response or adverse effects, and also evidence-based strategies for prevention and enhancing resilience, which we know far too little about. So this is where I'm going to say that precision medicine may be a way for us to begin to think slightly differently about what we do. And precision medicine is a term that may be morphed out of the notion of personalized medicine, which is a little bit of an older term, but this is how the Precision Medicine Initiative Working Group, which was convened by the government and ended up being the thing that launched this All of Us research program that Murray was referring to. This is how they defined it in their working group report. Precision medicine is an approach to disease treatment and prevention that seeks to maximize effectiveness by taking into account individual variability in genes, environment, and lifestyle. So in some ways, the key aspect of that is the individual difference and leveraging that. So, how might that apply to psychiatry? Actually, when I was going to give a talk about this subject, and I was meeting with a patient who said, where are you going next week? And I said I was going to give a talk about precision medicine. And the patient asked me, so what is that? And I said, well, it's, taking into account individual differences in people's lifestyle and, really understanding the individual. And she said, you mean like getting to know the person and really kind of leveraging their individual situation and trying to develop a treatment related to that? I said, yeah, that's exactly it. She said, wait a minute, what are you doing now? So that's a good question, because in theory, isn't that what we're supposed to be doing all the time? But the reality is that's not actually the way a lot of medicine goes. We are familiar with the concept of precision medicine as opposed to a one-size-fits-all kind of approach in a number of different areas. So the old school version of that is, eyeglass prescriptions. Obviously, you could go down to the local pharmacy and pick up a pair of reading glasses off the shelf, and they probably would help to some degree, but that's nothing near as good as getting your own eyeglass prescription and having it fit your particular needs. Blood transfusions, another very well-recognized and essential way that we individualize what we give to patients, because if we didn't, the results could be catastrophic. We need to type the blood, obviously. And in the last few years, there have been a number of really pretty substantial advances in the precision medicine framework in a number of diseases, not so much in psychiatry, but things like molecular typologies of cancer that have really reordered the way we think about hematologic cancers and some other cancers. Breakthrough treatments for certain conditions like new drugs for cystic fibrosis, which actually target the genetic lesion and have completely transformed the way we take care of patients with cystic fibrosis. Gene tests for drug side effects, which are now being used in areas of medicine. And the next frontier is going to expand on that and add things perhaps like risk prediction, new precision therapies along the lines of what I was just talking about, prevention strategies, new disease classification. So if we think about what are some of the major unsolved challenges in psychiatry, many of them are ones that I mentioned before. Some of the ones that we are working on in particular are things like, can we use genomics and other biomarkers to inform diagnostic classification? Can we identify individuals at increased risk of important outcomes like suicide or first episode psychosis or PTSD after trauma? Can we match treatments to patients? Can we use big data to actually identify and validate potential prevention strategies? So I'll talk about a few of those in the next few minutes. So here are some of the examples that I'm going to briefly touch on, and I won't, you'll see in each story there's much more to be done, but just to give you a flavor of the kind of thing that we're hoping to pursue. So the first one is something about clarifying the structure of psychopathology. And this is a thing that's well known to any of us in the field of psychiatry and mental health. That is that our current diagnostic system in psychiatry is an evolving story. So if you look at this graph from DSM-1 in 1952 to DSM-5 in 2013, and you've got this incredible progress in our coming up with labels for diagnostic condition. But we know that even with that, the labels, the boundaries, the classifications that we have are kind of, unstable. So if you just take the example of three kinds of conditions, pervasive developmental disorders, anxiety disorders, and mood disorders, and you start at DSM-3 in 1980 and watch what happens, you see that the diagnoses sort of come in and out, and there's a reorganization that happens. In DSM-5, actually, in some ways, there was a big transformation where all the pervasive developmental disorders congealed into autism spectrum disorder. We saw a number of anxiety and mood disorders either appear or disappear. And then we saw some of them move into whole new categories that kind of didn't exist before. So this is what we're kind of up against as we think about defining the phenotype and how these things are actually sort of a moving target. Despite that, we've had tremendous progress using these diagnostic categories in the field of psychiatric genetics, for example. So we've just seen, as many of you know, and some of you have led efforts in this area, the last decade has been really remarkable in what's been achieved in psychiatric genetics. And this sort of that takeoff starts around 2008 as genome-wide association studies, and also to some degree structural variation studies, took off. And now we just see this incredible inflection as we identify more and more specific genetic loci that are robustly associated with a whole variety of psychiatric disorders, schizophrenia leading the pack in this case. And that's for a couple of different reasons. But the point is, this is just an ongoing process. And what's remarkable about it is not only that 10 years ago we had practically nothing, and now we have a lot. Maybe some people think too much. But the fact that these are all grounding our biology. They are telling us something about what are the biological factors that are underlying disorder. And because these are what we call unbiased genome-wide association studies, mostly it's new stuff we hadn't thought about before. So these potentially are giving us new leads into the biology, potentially new targets for developing therapeutics, and that's enormously exciting. The other theme, though, or one of the other themes that's emerged in the last few years is that, surprise, the genetic basis of these disorders doesn't neatly map onto the DSM. It's not as though there's a genetic variant that's associated with, or a set of genetic variants that are just associated with schizophrenia. There may be a set of them that are, but by far, the story appears to be one in which there's a lot of sharing at the genetic level. There is a lot of crossing of our diagnostic boundaries. One of the largest efforts came from the Brainstorm Consortium. Most of the work in this area is from very large consortia because you need these kinds of powers, sample sizes to get the power you need. This was a study that put together genomic data for a whole bunch of psychiatric disorders, neurologic disorders, and actually other traits. And what you're looking at in these boxes and these grids are the genetic correlations, so the intercorrelations at a genetic level of these disorders. And I don't know, does this pointer work? Yeah. So for example, in the psychiatric disorders, you've got ADHD and its correlation with a whole variety of disorders here. Same thing for anorexia. What you see is the darker blue means more deep genetic correlation. There's a lot of blue. Not so much blue among the neurologic disorders, and very little actually between psychiatry and neurology, which is interesting. It kind of goes against our ethos of these are all brain disorders, and they might, they are all brain disorders, but at a genetic level, we don't see that degree of overlap that one might have perhaps expected. But there's some pretty substantial genetic overlap between some of our disorders. Bipolar disorder and schizophrenia has a genetic correlation of about 0.7, so 70%, you could think of it. That's amazing because those are two conditions which are kind of the canonical diagnostic distinctions in our field. Emil Kraepelin and everybody else, that's what we kind of hung the whole system on, being different. They're mutually exclusive conditions. You can't have both, but they're genetically incredibly closely related. The largest effort so far to kind of look at excuse me, the locus level and to maybe broaden this out to some degree has come from a study that the Psychiatric Genomics Consortium's cross-disorder work group recently published. Actually, before I get to that, this is a really other interesting example, I think, of how looking at these genetic relationships may give us new clues into the underlying biology. So this is actually a study that looked at, again, from PGC investigators, looked at the relationships of the genetic basis of anorexia nervosa to other conditions. And one of the findings that emerged was not only is it related to a lot of other psychiatric disorders at a genetic level, it's also related to a lot of metabolic phenotypes. And the comment in the paper was, just as obesity is increasingly considered to be both a metabolic, endocrine, and psychiatric disorder, approaching anorexia as both a psychiatric and metabolic condition could ignite interest in developing or repositioning pharmacologic agents for its treatment. This has been followed up with another even larger analysis that confirms these kinds of relationships. So it does tell us something interesting, perhaps, about how we might think about the relationships among disorders. And as I started to say, the biggest of these efforts recently from the cross-disorder group, analyses led by Phil Lee, put together data for 8 psychiatric disorders: anorexia, ADHD, autism, bipolar disorder, depression, OCD, schizophrenia, and Tourette syndrome. And the first thing was to look at the relationships, again, among these disorders at a pairwise genetic level. You can't really read these numbers on the lines between these nodes, but here are the individual disorders. And the strength and width of the line tells you something about how closely related they are. So once again, we see, and also their position. Schizophrenia and bipolar disorder are very closely related, but also pretty much all of these things were pretty closely related. But then we were able to use another method called genomic structural equation modeling, which is a way of actually trying to find structure, not just at a pairwise level, but if you just took the genomic data and looked at its structural relationships, what would you see? And what emerged there was something really interesting, which is that if you put all those genetic data sets together, you get these three factors of more closely related disorders. So the first one included what you might think of as compulsive behavior disorders, or disorders that have a compulsive behavior component, so anorexia, OCD, and Tourette. The second one were mood and anxiety disorders. The third one were sort of more early onset neurodevelopmental disorders, so autism, ADHD. There was also a component of depression, maybe that was an early onset subgroup or something, and Tourette syndrome as well, loaded on that factor. So that's kind of an example of how using genomic data might give us some insights into how we might think about classification. It's not certainly enough to change our classification, but it illustrates the kind of thing that we could perhaps do. We also did a, excuse me, I get choked up when I see these results. We also did a genome-wide association study meta-analysis and found 109 loci that were associated with at least two disorders. And what you're looking at here, probably people are familiar with this kind of a plot. This summarizes the results of a genome-wide association study. It's called a Manhattan plot. And basically, you've got the chromosomes lined up on the x-axis. Each of these little dots, which coalesce into just blobs down here, are markers, common variant SNP markers. And then the height that they are on the y-axis tells you something about their statistical significance. And this is given as the minus log p-value. So 10 on that scale is a 10 to the minus 10th p-value. And then, this line is the statistical significance threshold of 5 times 10 to the minus 8. So these are all loci, and in some cases mapped to genes that you can see here that were associated with more than one disorder, what we would call pleiotropic loci. And some of them were remarkably pleiotropic. So the most pleiotropic was a gene called DCC, which stands for deleted in colorectal cancer. Sounds odd, although that also turns out to be a master regulator of white matter patterning in the brain during development. So now it's not so weird in a sense. And that was associated with all 8 disorders. I won't go through the details of this plot in the interest of time. RBFOX1, another one that had emerged in some prior genome-wide studies, also associated with a very broad range. And then you can also look at sets of genes or biological pathways and see if you took all these genetic results, what are the pathways that seem to emerge as containing more of these results than you would have expected by chance? And what we saw was that these pleiotropic loci were enriched for neurodevelopmental processes, so neurogenesis and regulation of nervous system development and CNS neuron differentiation. Also, a couple of specific neurotransmitter pathways, particularly glutamate and calcium channel signaling. And then we could do functional genomic analyses that also were rather interesting and suggested that these more pleiotropic, that is these loci that have, or genes that have very broad effects, seem to show heightened expression in the brain, based on postmortem gene expression studies, that begins in the second trimester, so this blue line, and it stays high. Whereas the loci that are associated with one disorder sort of had a similar, or rather an opposite pattern. There was marked enrichment in frontal cortex when you look at cell types, and preferential expression, especially in cortical glutamatergic neurons in layers 2 to 6. So we can begin to tease out a little bit of the biology as well. Okay, I'm going to shift now to another story, but that sort of gives you a little bit of a flavor of how genes could inform the way we think about our diagnoses. The next thing I talked about as an area of interest is risk stratification. And the particular issue that we're very interested in and trying to pursue to a great extent is suicide, which of course is a rising problem in this country. More than 1.5 million people make a suicide attempt every year. More than 47,000 die, which puts it at a higher death rate than many of the things that we hear much more about. And there has been this sort of steady increase in the rates of suicide in this country. It went up another close to 2% last year. It's the second leading cause of death among young people. And, you know, most people, or maybe half the people, don't actually have a known mental health condition, which suggests that, you know, we may not even be able to use the fact that people are coming for help to know. So one of the things that we've been excited about is the opportunity that's provided by big data and AI or machine learning approaches, where you can take very large data sets that are now available and try to extract information and incorporate potentially more information than any human being could do. And you're not relying on self-report, as I'll tell you a little bit more about this. So one of the problems with suicide in particular, obviously, is that sometimes people are motivated not to report about their intent. It also turns out that most people who attempt or die by suicide are seen by a clinician in the months leading up to it. So we have an opportunity in the health system to begin to address this problem. And that's what we've been trying to do. So one of our first forays was to build a model using electronic health record data, which is, you know, ubiquitous. You guys have Epic here. You know, almost all health systems have a pretty large electronic health record system. We certainly do. We queried our electronic health record system of more than 4 million patients and built a model using a machine learning approach, which is a fairly straightforward one called a naive Bayes classifier, which essentially treats every feature or every variable as an independent predictor. And we had about 15 years of longitudinal data, 9 million person years. So it's a pretty big data set. The nice thing about electronic health records, and there's some not nice things about them, but one of the nice things about them for research is that they're constantly accruing and just passively growing. As we're sitting here, thousands of observations are being recorded in the electronic health record. We identified about 17,000 cases and many more people who were not cases. After reviewing charts and identifying definitions, we added in death certificates, and we trained a model in 2/3 of the data and validated it in the remaining third. And this is a measure, the AUC, the area under the receiver operating curve, which is standardly used to assess how good at discriminating cases from non-cases something is. And we like to see, so the diagonal line means it's a coin flip. It's no good at all. You like to see something in the range of 0.7, 0.8, or higher would be even better. And so this did actually pretty well. What this is plotting is the sensitivity, how good it is at detecting true cases, over the false positive rate, which is 1 minus the specificity. So in the end, this model detected 45% of all attempted or completed suicides at 90% specificity, an average of 3 years in advance. And one of the things that is interesting about this is that it, again, picks up things that you as a clinician might not be thinking about as a predictor. So on our list of top predictors, and they're, you know, this is a model that actually uses everything, were things that you would predict, like, you know, substance use and mental health conditions, but also these things like hepatitis C carrier, alveolitis of the jaw, osteomyelitis, a bunch of infections, actually, which were interesting. So we were encouraged by this, but we also wanted to know, could this work on a larger scale? Would this work in other healthcare systems? And so we tried to do a study to see if the approach that we took of training this model, validating it, could work elsewhere. And we collaborated with a network called the ARCH network, a research network that linked electronic health record, linked health systems with electronic health records at 5 other institutions. And basically what we did was we used the same approach at each of these institutions, which are spread out around the country. They have very different patient populations. One of them is a pediatric-only hospital, Boston Children's Hospital. We trained in 50%, tested in the other 50%, and then looked again. And the results were pretty similar to what we had originally found. So about 40% were detected about 2 years in advance at 90% specificity. The AUCs were in that same, 0.7 to close to 0.8 range. The top decile of risk captured about 33% of the cases. And there's a lot more work to be done on this, and we can get into the nuances of it. But thinking ahead, one of the things that we're trying to do is now develop a clinical decision support tool. It is possible to develop essentially apps for the electronic health record that can be used at the point of care. And we're prototyping and developing these things. We're doing lots of focus groups with clinicians to ask them about when would you use something like this? What do you worry about? And we're also planning a trial to test the value of giving this kind of information and coupling it with a suicide risk prevention consultation team and a set of interventions that we can use to assist primary care physicians, probably predominantly. So that's an example of a story on risk prediction. There's also the question of, can we do better by combining genomic and EHR data or using genomic data in a health system? And for that purpose, along with Leah Davis and folks across the country, including Murray, we have established a consortium called Psych-Emerge, which builds on something called Emerge, which is an NHGRI-funded consortium of electronic medical records and genomics. That's where the Emerge comes from. And basically what we've done is we have formed a coalition of health systems and resources where we have electronic health record data linked to genomic data. And it turns out that is a growing kind of thing. So we now have EHR data for more than 12 million individuals and genomic data for more than 1 million if you put everything together. And these are the participating sites currently. One of the things that we've looked at in this context, especially on the genetic side, is not looking at individual variants, because it turns out this is not all that well suited to discovering new variants for reasons, again, that we could talk about. But there is another sort of theme emerging from genome-wide association studies, which is that at a individual and maybe personalized medicine or precision medicine level, what could be useful are two kinds of genetic variation for either diagnosis, prognosis, et cetera. One of them are rare, highly penetrant copy number variations, which are big chunks of DNA that are deleted or duplicated, which we all carry, or mutations, but those are rare. And the other is more common variation, but not these single variants, actually putting them all together in what we call a polygenic risk score or a genetic risk score. So whereas individual SNP variants have very small effects, if you put them together, you actually can capture a lot more of the variation. And this is just the simple formula for a polygenic risk score, which is that it is the sum of the effect sizes. So you do a GWAS, every SNP gets an effect size, and at every SNP, you have all the SNPs, each of us in this room, you have either 0, 1, or 2 copies. So we take your 0, 1, or 2 copies at every SNP times the effect size, sum them all up into a single score. And that single score is kind of an index, to the extent that we can do it, of your common variant genetic liability for whatever it is that score was trained for. And it's normally distributed. So some people are at the tails, but most people are in the middle. So we wanted to know that those polygenic risk scores have been increasingly looked at. And what they show is that, for example, for schizophrenia, you can robustly train a polygenic risk score in one sample and show that it's very strongly associated with schizophrenia in another sample. And that's great. And if you look at the effect size of that, if you take the people at the top 10% and compare them to the people at the bottom 10% of that risk distribution, they have maybe a 8 to 20-fold increased risk, which is not nothing. But the question is, what is that like in the real world? Because those are really rarefied research samples. Would this work if you were actually thinking about bringing some of these things to medical practice? Could this possibly work? And what we did through this Psych-Emerge consortium was put together data for 4 different healthcare systems, more than 100,000 patients, And we took these polygenic risk scores that have been trained for schizophrenia by the Psychiatric Genomics Consortium, and we again found that it was strongly associated with schizophrenia or psychotic illness in a real-world healthcare system setting, using very kind of minimal phenotyping. The odds ratio was not as high as has been reported. So if you took the top versus the bottom decile, put these together in a meta-analysis, it's about a 4.5-fold increased risk. We used a method that Tian Ge in our group had developed that actually supercharges the polygenic risk score a little bit. And so this was the top versus the bottom decile. The top decile versus the other patients is about a 2.3-fold increased risk. This was led by Amanda Zoitland, who was a postdoc in the lab at that point. So you could take a couple of things away from this. One is we see in the real world that these polygenic risk scores work. We also see that they have a moderate odds ratio. Now, that odds ratio is not that different from odds ratios that we use in risk calculators in other areas, like in the Framingham risk score for heart disease, you know, the cholesterol, hypertension, smoking, they all have about that effect, and we feel kind of comfortable with that. So one thing you might imagine is, what if you combine this with electronic health record data? And we're working on that now, and this is a very much in progress story, but a first look that Amanda did was take that naive Bayes classifier approach, trained it, and then added in people's PRS decile risk. And the bottom line here was the EHR swamped the genetic risk. So its AUC was 0.91, which is amazing. It was being in the top decile was associated with a 28-fold increased risk. If you add in the PRS, it doesn't do much. But that's clearly only part of the story. It depends on how you add in the PRS. And also, if you have years and years of data on people, that's a lot of information. But what if somebody's just coming in with a first episode, or maybe not even a first episode? Maybe then the genetics actually could contribute more. I'll just mention the other thing people often think of with precision medicine, precision psychiatry, and that is pharmacogenetics and tests to guide treatment. That's a huge need, right? Right now, if you see a patient, and let's say they have depression, you're kind of winging it in terms of which medicine you're going to start with. There's some, folk wisdom about some type of medicine might work better than another, but pretty much we're doing guesswork. It's trial and error, and an enormous amount of Americans are on psychotropic drugs. So this is a big problem, and every time you're wrong because they couldn't tolerate it or it didn't work, you have to wait weeks to know the answer for that. And meanwhile, people are suffering, maybe they're not able to work, et cetera. So there are also, adverse effects that can be catastrophic for some of our medications. And we know the treatment response is partly heritable. So this is like an enormous need, and it ought to be doable. And I'll just cut to the chase about, I think, where I think the field is, and that is it's not very well doable at this point. So there are many commercial tests available, and doctors and maybe people here are ordering them, and some of them are covered by insurance. Mostly they use what we call pharmacokinetic targets, P450 enzyme metabolism variations, and then a few others thrown in. The largest sort of controlled trial used the GeneSight test. Didn't make, it wasn't significant on its primary endpoint of symptom improvement, but did show effects on response or remission. My take is that I think it's still unclear when these tests are helpful and if they're helpful, but I'm hopeful that we're going to see more evidence. And they don't have to be perfect, because right now we're on that diagonal line of a coin flip in many ways. Okay, let me switch to a third story. I think this is the third story. And that is the notion of using genomics to identify promising prevention or intervention strategies. That sounds weird, but I'll tell you what we mean by that. First of all, what do we know about preventing psychiatric disorders? Let's say depression or schizophrenia. What can you do? Well, don't have affected first-degree relatives. Don't have significant childhood adversity. and just say no to drugs. Those are pretty much the big contributors that we know about, the big risk factors. And there's not a whole lot of, I mean, the third one, I wouldn't put it that way, but maybe we can treat or prevent substance abuse. The other two are kind of hard to tackle when somebody comes in, for example, because they've happened already. In the realm of depression, there are a couple of things that have been suggested to have preventive effects, and those are physical activity and social support. And there is a literature about these things. So one of the studies, and these have really been led by Carmel Choi, who's a postdoc, senior postdoc in the group, and she was interested in, could we find evidence that physical activity actually is causally associated with a reduced risk of depression? And the reason for that being a question is, of course, if you see an association between physical activity and depression, that could be because physical activity, more physical activity, less depression, maybe it's preventive. Or if you're depressed, you're not moving as much, or even if you haven't been diagnosed, and that's just a reverse causation. So she used a technique which is increasingly being used and may be familiar to you, called Mendelian randomization. And the basic concept here is that if you have genetic variants that are associated with an exposure of interest, so let's say you have an exposure, typical epidemiologic kind of question, and an outcome. And you want to know, is the exposure related to the outcome? Could be related to the outcome because it's confounded by something. But you want to know, is it causally related to the outcome? One way you do that is through a randomized controlled trial. That's what we do in drug studies, right? You effectively randomize people so all the other confounders are roughly equal, and then you just give them a treatment or not. And now you can make a causal inference, because that's kind of the only real difference. You can't do that with certain things that, certainly in observational studies, you can't really do that. So Mendelian randomization effectively tries to, you can think of it as effectively trying to randomly allocate people to higher or lower levels of the exposure based on their some genetic factor, which you have previously shown is associated with higher or lower levels of that exposure. And then you want to know, is there a relationship between this genetic variant, which affects this exposure, and the outcome? And the formula is sort of deceptively simple. So you're trying to estimate this beta, this effect, and it's the ratio of this effect, the effect of the genetic variant on the outcome, over this effect, the genetic variant to the exposure. Allows you to make a sort of causal inference, and you can actually do it in the reverse direction. So you could switch the exposures. Depression, does that cause physical activity to be lower? She did this using the UK Biobank, which is one of these big resources that is now available, 500,000 people. And it turned out in the UK Biobank, not only did we know who developed depression, but people had worn accelerometers, like your Fitbit, to measure your physical activity level, about 100,000 people. So she could ask this question with objectively measured physical activity. And the answer was, there was a causal protective effect based on this kind of analysis. About an odds ratio of 0.74 for each standard deviation increase in average acceleration, which translates to about replacing 15 minutes of sitting with 15 minutes of running, let's say, or an hour of sitting with an hour of moderate activity. There didn't seem to be a reverse association causally in this analysis, which is interesting. Carmel subsequently did an analysis using our own biobank of partners, in which we had, again, people's, this time it was self-report, but their estimates of physical activity, and then we could prospectively look at who later developed depression. And we had genetic risk scores now. So we could ask, does it depend in some way on your genetic liability to depression? So the first thing she found was that more physical activity, less incident depression. But encouragingly, it didn't matter what your genetic risk score for depression was, the protective effect of physical activity was about the same. So that's pretty robust. The other thing I mentioned was social support, and through the Army STARS program, which Murray has led, we were able to look at at this question in an interesting way, which was that we had soldiers who had been deployed to Afghanistan and who were studied prior to deployment and after for, let's say, the next 9 months to see who developed new onset depression. And there is a concept in the military called unit cohesion, which is kind of a measure of how much you feel like your unit members sort of have your back, your superior officers can be trusted, you feel connected to your unit, you guys and women are all together in one unit. So we had about 4,000 folks of European ancestry, which was the restriction for the genetic analysis. We could ask, to what degree does genetic risk predict incident depression after returning from a defined, pretty stressful experience? And how about the degree of the combat exposure or trauma exposure? And what we found was the genetic risk scores actually did have a dose-response relationship to incident depression. More genetic risk, more likely to develop depression after combat exposure. Unit cohesion had an overall very protective effect, which was really interesting. And then we could ask, does that work even for people at high genetic or environmental risk? And the answer was yes. So even for people at high polygenic risk, that protective effect was there. And even for people who had high levels of deployment stress, that protective effect was there. So in both of these cases, actually, physical activity and social connectedness, let's call it, these things seem to be protective despite your underlying risk. So more recently, Carmel has taken this to a next level and said, okay, so physical activity, social support, what if we looked at all of the potential modifiable factors we could find? And so using, again, the UK Biobank, which has this very large data set, asked, let's identify the modifiable factors. We found 105 potentially modifiable factors, and then did the kind of analysis that we just talked about. But here, the interesting thing was to say, okay, we know who's not depressed at baseline and who then later gets depressed. Now let's look at the association of any of those modifiable factors with getting or not getting depressed later. But that's an observational framework. So then the strategy was take the ones that are associated and now bring them into Mendelian randomization to say, are they causally associated? So it's kind of a two-stage approach. So here's what she found, and I realize you can't really read this, but I'll tell you what it says. This is a kind of Manhattan plot of the modifiable factors and their effect on incident depression. You see one thing screamingly positive up there, although anything above this line is statistically significant. I don't know if you can read that, but that is frequency of confiding in others. That was very much associated with a reduced risk. And here are a bunch of factors that showed some effect in different domains: physical things, media, circadian rhythm kinds of things, social, environmental, dietary. And then the ones that were significant, she brought into this Mendelian randomization. And the answer there was that there were a few things that were significantly showing a causal protective effect, and similarly, a few things that were showing a causally risk-increasing effect. Excuse me. On the risk-increasing side, TV use, which might be a proxy for either TV use, or it's a very good proxy for TV use, or other kinds of screen time, or maybe sedentary behavior. Daytime napping, which again, on the protective side, frequency of confiding in others has that effect, and it appears to be protective. Also, frequency of friends and family visits, exercise, a couple of weird ones. Multivitamin use had a negative effect. I don't know. It turns out there is a little bit of a literature suggesting that that might be true, but salt intake also. Anyway, you can see this strategy, and it's a way of using these kinds of things to inform prevention. Lastly, I'm going to tell you about another kind of approach, which is genome-guided therapeutics. There's actually a literature that shows that, we know that the cost of developing a new drug is massive, and most drugs that enter the pipeline crash and burn, and that's why the cost is in some ways massive. But it turns out that if you have a drug target that is genetically validated, that is, it shows evidence from genetics of being associated with the outcome, that doubles your probability of succeeding. So there's a lot of interest in this kind of thing. The notion is you would, start with a genetic association. There's still a long pathway to getting a drug, but you're on maybe more solid ground because you have grounded the drug in human biology. And then you have to do all the things that you do. Sometimes you get lucky and you can repurpose a drug that already exists. So one of the, in studies that are led by Robbie Mueller in our group, who was a postdoc, now a junior faculty member, he's been looking at a gene called SLC39A8, which actually turns out to be the strongest, it's a coding exon functional variation, changes the amino acid sequence, and it's not that rare. So that itself is a rare phenomenon. And it has emerged strongly as a risk factor, a variant in that locus, in schizophrenia, from the schizophrenia GWAS. And this is a missense mutation in this gene, which turns out to be a manganese transporter. Manganese, a mineral or an element that is an important cofactor for lots of enzymes. This variant itself is present in maybe 15% of people of European ancestry. And manganese is a cofactor particularly of interest for glycosylation reactions, the addition of sugar moieties to proteins and lipids. And that's really important for a whole series of brain developmental processes. There are severe congenital deficiencies of this gene, where the gene is essentially knocked out. And those cause congenital disease of glycosylation. And that's associated with severe developmental brain abnormalities, undetectable manganese levels in the blood, and impaired glycosylation of transferrin, which is just an example of a protein that gets glycosylated. And interestingly, that gets reversed by just giving these people manganese. Or galactose, which is an intermediate in the glycosylation pathway. So one of the things that Robbie did was ask, well, I won't go into this in detail. There's been a little evidence that maybe this variant is associated with differences in basal ganglia brain structure, caudate, putamen, and with a collaboration with our other colleagues at MGH, we found that that's probably not the case. It's probably a difference in signal that results from the fact that you've got a divalent cation, as iron is, for example, that is altering the signal in the MRI. I won't go into greater detail on that. But one of the things that we can do now with a biobank is actually call people back or call their samples back based on genotype, because we've got genotypic data on everybody in the biobank. So we could say, let's find folks who carry this variant and people who don't. And what Robbie was able to do was to look at, first of all, their serum manganese, if this really is having an effect like we see in these more dramatic cases. And he found that, indeed, carrying the variant was associated with a lower serum manganese. And then he used fairly sophisticated technology that allows you to look at the glycome, this sort of array of glycosylated proteins, in particular the N-glycans or N-glycome, and found that carrying this variant is associated with a more a reduced branching of these sugar structures on proteins. And the reason that makes sense is that if you don't have the cofactor, and this kind of adding sugars is a sequential process, you never get all the way to where they need to be in terms of the complexity of the sugars on the molecule, on the proteins. And so this seems to be a consistent thing where you carry the variant, you have less manganese around, your glycosylation machinery is essentially messed up. He was able to also have the blood from the cases of severe congenital disorder and found the same thing. That is this kind of more primitive branching. And again, in the interest of time, I won't go into the details. And one thing that we're hoping is that this might provide an interesting therapeutic opportunity, because abnormal glycosylation can now be targeted based on the evidence of these prior studies with oral supplementation. So could it be that actually giving manganese or glycosylation precursors could have a therapeutic effect? I was talking to Neil about this yesterday, and a major threat to this is that the problems may have already occurred in development. And so maybe that's not so reversible at a phenotypic level, but we're going to find out. So let me end by saying, is precision psychiatry a thing? I think it's becoming a thing. That's my bottom line. So we're beginning to see application of precision medicine approaches and tools to psychiatry. There is an urgent need to do this and to do better than we're doing. There's some early encouraging results, but there's still a long way to go. We need to capitalize on some of the resources that are now available that weren't available a few years ago, large-scale data resources, integrating psychiatry, genomics, epidemiology, neuroscience, and implementation science. Everybody gets a job in this new future. And we also do have some resources that are pretty remarkably helpful. I showed you a couple of these, but these biobanks linked to electronic health records where people are call-backable, if that's a word, are potentially incredibly powerful resources. There's so a lot of institutions have these. There's the Million Veteran Program, which Murray is deeply involved in and has led a lot of analyses in, which is, 1 million veterans. One of the nice things about the name of that is it tells you, as opposed to the All of Us program, which is another million-fold study that is particularly interesting because it has representation from many communities that have been underrepresented in biomedical research, which is a huge problem in the field. In Europe, there are these incredible resources that have been collected, and in Asia as well. So there's a lot of work to be done, but there are now a lot of tools and resources to do it, which I think is wonderful. So I'll just end by thanking the many people who are involved. This is just some of the folks involved in that cross-disorder work that I mentioned. The electronic health records prediction work, and the PsychEmerge consortium as well. And of course, our funders, viewers like you. So thank you. Thanks for listening, and be happy to take any questions. Thank you.
Wow, I think if anybody asks what makes a great Grand Rounds talk, if you can work EMRs and glycosylation into the same talk, and if anyone asks what makes psychiatry so daunting, it's that the material we're dealing with spans that whole level, goes from manganese to how many people you can confide in, whether you eat lamb or multivitamins. It's a daunting process, but thanks for the A to Z description there. Questions for our speaker?
Yes, quite impressive, thanks. I remember Tom Insel came out here many years ago and told us how we hadn't budged suicide rate. And then subsequently, Myrna Weissman's group at Columbia came out and found that for those individuals who rated spirituality as important to them, it reduced their risk of suicide by 90%, as well as that of their offspring, irrespective of the views of the offspring. And it was associated with changes in white matter connectivity measures of the brain over 20 years. And now that finding has been supported by data out of the Harvard School of Public Health and the Nurses Healthcare Study. I'm wondering how you think that effect may be mediated. Is that through confiding in others, or is there a connection there, or social support, or? Really
interesting. Yeah, those there have been a series of studies of spirituality, also, religious attendance in organized religion as well, but spirituality is a broader concept. Do seem to have, are consistently, or in many studies, seem to be associated with reduced risk of a number of outcomes, not only psychiatric, but cardiovascular and that kind of thing. I have no idea, really, what is happening there, but you certainly could imagine a number of possibilities. One of them being this kind of sense of community that often goes along with being part of a spiritual community, or even in a certain spirituality of a connectedness that's not to other people, but is connected to something. And whether we can go all the way to the biology and say it's changing the brain in some way, I think we're probably far from knowing how that would be possible. But the effects of spirituality and those kinds of deep experiences, the capacity to think transcendently, all those things might have their own, at a psychological level, protective benefit. I don't know, but it sounds like it's a real possibility.
Yeah, I mean, I just remember seeing people in the intensive care unit, young people, no risk factors, but had no sense of the meaning and purpose in life, and made really serious
And when I see patients and
attempts.
I'm worried about them, I ask those questions because that's a gauge in some way about, occasionally it goes in the opposite direction where people feel like, I know there's a better life after this, and that makes me worry a little bit.
I also want to congratulate you for the talk. It was really stupendous.
Thank you.
One of the advances in precision medicine has been in cancer, as you know, and gave in the beginning of your talk. But that's tissue typing. That's not germline profiling.
Right.
And I think this relates to Barbara's question a little bit. So what do you think in psychiatry about the possibility, could we use white cells, could we use other tissues that might reflect changes in the brain that would give us some idea of chromatin remodeling, methylation, RNA-seq kinds of things? And could some of these psychosocial issues be changing those more than they're obviously changing germline?
Right. That's a great question. I think there's a lot of, with good reason, excitement about the possibility of looking at these kind of other omic markers and the possibility that, for example, the environment and experience may shape things like DNA methylation, chromatin remodeling, that itself shapes gene expression, and et cetera. It's a little daunting because one of the things we know about, for example, the epigenetics is that it is pretty tissue or region specific. Sometimes there's appears to be some overlap with certain brain regions, but by definition, in some ways, we're using a peripheral echo of something that may not be really going on in the brain. One of the things that I found fascinating talking to folks here yesterday was the possibility of using exosomes, which are these little packets of cytosol that capture proteins from different cell types and are now circulating in the blood, so you can get neuronal or glial exosomes, and maybe that would be a way into actually seeing what but it's not giving you DNA methylation really, or at least could be, I suppose, but it would be unusual, but can give you microRNAs and proteins and things like that. So I don't know, I find it daunting to think about, so the way that you could get these sort of more peripheral things and modifications that are not necessarily inherited from blood markers, white cells, or others, for DNA methylation, you'd be saying, for whatever reason, maybe it's not exactly what's going on in the brain, but it's reflecting, it's a proxy for something that happens to be correlated with, let's say, responding better to this treatment than another treatment. And I think that's a real possibility. Haven't seen it yet, but that would be one way that you don't have to hang your hat on it being the actual thing that's going on in the brain. But you could, through data mining, essentially, show that if you have this series of, it could be modifications to the DNA, it could be serum proteins or that change, or metabolites. I think it's going to be a complicated thing. You're right, we're very much disadvantaged relative to cancer in not being able to get the tissue type and look.
In animal studies, if you have brain tissue and you have blood, then you can compare.
Yes.
And by doing that, we may have a subset that then we can look at in humans.
Right, and that has been done.
And then also, if we start collecting brain tissue when people
Yes.
are doing
Surgery.
surgeries,
Yeah.
then we may be able to develop a tissue bank
That's
can be
right.
used for that too.
That's right. Yeah, I think that's very, very promising. John.
Great talk, Jordan.
Thank you.
I just have a very kind of practical question. In our early attempts to use the EHR, we discovered that psychiatrists are allergic to numbers. So in other words, extracting any kind of quantitative data out of progress notes, which is our primary source of data, was just very difficult. I was just wondering how when you said we're going to put the whole EHR into a naive-based classifier, well, what variables, because that's the hard part, how did you extract variables from it?
Right. In this case, what we did, although we've done it differently in other cases, in this case, what we did, we used the structured data only, meaning diagnostic codes, prescriptions, labs, procedures, and then you don't, so you're missing a lot. What we've shown in other studies is if you use natural language processing, which can actually extract and turn into essentially binary or other variables, the text itself, by parsing the text, using ontologies that can map what's in the record to defined concepts, you can increase the sensitivity, you get a lot of additional information. And we've done that now in a couple of cases in bipolar disorder. For example, we trained a model that used NLP, and we compared it to in-person blinded diagnostic interviews using the ScD, and it did really well. I mean, its positive predictive value was close to 0.9. Its genetic correlation with PGC bipolar data was about 0.83. Seems to be really capturing something real. But it's a more intensive thing to do. The reason we did it this way here was we wanted to maximize the kind of portability and computational efficiency of it. So that is a challenge. I mean, you know, the EHR has a lot of great things about it. It has a lot of terrible things about it. For example, if you use a single diagnostic code, ICD code, as your phenotype, we now know from a whole bunch of disorders, especially in psychiatry, that the positive predictive value of that is the chance that if you have one code, you have the disorder, is about 30%. It's terrible. So you really have to be careful about how you build these definitions.
Yeah, I really like the talk a lot, and I'm particularly struck with the confiding in others business, because isn't that what you guys do, you clinicians do? It's like people come and they confide in you, right? That's my vague understanding. Obviously, without training, I could completely have misinterpreted it.
I
but,
think, technically,
so, But so are those interchangeable, right? Is it the case that in the absence of unit cohesion, if you had an outside person who was some kind of a therapist that people could confide in, that would be a appropriate surrogate? I don't imagine you know the answer from the data, but doesn't that seem like an important...
It does, yeah. No, that is a really good question, and you're right. I mean, you know, people used to talk about therapy as a corrective emotional experience, right? And that you, maybe there is. How would you test that? I guess you would, I mean, it's certainly the case that there's evidence that psychotherapy, which involves confiding in others, helps people. But you're right, it would be interesting in the, I don't know if you could do it in STARS. Do we have social contacts outside of the military? Not really. But that would be a really interesting question.
I also kind of suspect it's similar to the loneliness concept that, you know, Sandra and I have thought about.
That's right.
I think it may be just a different angle.
Yep.
Yeah, so maybe more the propensity of somebody to be willing to confide more so than the opportunity to have someone to confide in, or maybe it's both. So my question, I was taught long ago, and our department actually was built around this in some degree, is that to really understand the biology of a system, you need to push it. That static markers, single point, or genes, proteins, etc., aren't as informative in understanding the specifics or of the individuality of the system as what you see in a delta, a change from stressing the system. So cardiac treadmills, glucola tests, medicine is full of these opportunities to push the system. And here, long ago, we did physostigmine challenges in depressed patients to try to push the system. So my question is, built into this battery of potential markers for precision medicine, there's many that are static. Are there ones that we could explore that involve sort of pushing the system to make it identify and define its lesion?
That's a great question. And I agree with you that sort of probing, challenging, and seeing how the system responds is a key area of importance for probably identifying markers and things like that. So there are, for example, not emerging from this, but there's work in, evoked response potentials and its differential ability to predict treatment outcomes. Same thing with functional neuroimaging in response to task-based functional neuroimaging. But then there are also things like, so have I seen any of those? These possibilities certainly exist, including things that could challenge the HPA axis or the sympathetic nervous system and have readouts like heart rate variability or other things. I haven't seen, but I just probably don't know, anything that has been, except there was this recent paper, I think in Nature, right, about EEG prediction of treatment response. But I think that was resting, not ERP, but I'm not sure about that. So that kind of thing, I think, is really potentially valuable. When people do cellular assays or, animal assays, those kinds of things are, first of all, maybe easier to do and also tend to be very informative, I think.
Right. It's harder to get out of the electronic records,
Yes.
such a thing, because you require 2 time points and some controlled intervention between.
Right.
Sorry, I didn't mean to take control. Any other questions?
Hi, thanks again for the great talk. I have a question. I'd just like to hear your thoughts about how you might take this conceptualization of genetic disease where everything is sort of related to everything and translate that back into a practical diagnostic system that's better than DSM-5. I mean, I know that this is a hard question, but I'm just kind of curious about how you would go about thinking about that problem.
That's a good question, because there's an important issue there, which is the fact that things share risk factors doesn't make them the same thing, right? So, we see that genetically, for example, ulcerative colitis and Crohn's disease are highly genetically related, but they're somewhat different diseases. Lung cancer and COPD, their primary risk factor that accounts for most of the variance is shared, but those are very different diseases. So I wouldn't want to think that, I wouldn't want to leave the impression that I think, we're just going to collapse all these things by their genetic relationship and have a better than DSM, because the DSM is not purely about the underlying biology. RDoc has taken this approach, but if you think about it, the reason that we find something like the DSM useful is because we can answer people's questions about is this medicine likely to work for my general category of thing? What's the prognosis likely to be, et cetera. And we're not anywhere close to that yet on a bottom-up biological level. So I think it's not a trivial thing to do. I'm encouraged by this kind of thing that emerged from these cross-disorder studies of being able to look at genomic relationships in a way that maybe doesn't reorganize classification, because it's not enough on its own, but maybe organizes our thinking about the biology that might lead to treatments or to developmental timescales for different disorders that are related. As part of a, I don't know what you would call it. It was an intensive effort a couple of years ago that I was part of, and a number of other people. We sort of developed a Bayesian framework for how you would iterate information that comes from a variety of sources to improve and test repeatedly the classification system that you have. And what emerged from that is that it is possible to do that. So if we really thought about it in a kind of systematic Bayesian way, and the field took some effort to do this, and Josh Gordon and others have also written about this, I think it is possible for us to go beyond simply the consensus of experts approach that we have, but it's not trivial, and utility is, clinical utility is a criterion that's a very important one to keep in mind. So I'm not in favor of throwing out the DSM, but to the extent that we could incorporate things that we know, That's how for whatever you think about it, that's how the reorganization of the traumatic stress disorders emerged. And I don't know, is that a good thing? You probably were involved in that or something, right? But they were pulled out because they're not quite the same biology and course as, generalized anxiety or phobias. It'll be kind of that kind of thing, I suppose, but maybe informed more by these other data sources in a more evidence-based way.
For the last word?
Question. So Mike was sort of asking about how the fact that these disorders seem to be strongly genetically related, do we just kind of have one big psychiatric disorder? The answer is probably no. Mm-hmm. But how does this kind of work lead to taking even one category, major depression, and we all sort of always say, oh, there's more than one type of major depression. How do these types of data get us to being able to stratify even within disorders?
Right. Well, people are taking different approaches to that. Some of them are kind of unsupervised, if you have a big enough data set with enough variation to look at and then validate with various criteria, structure within a diagnostic category. What's the heterogeneity? And there have been a number of attempts to do this with schizophrenia, with depression. I would say that none of them have established subtypes that have yet borne out that are, because you need a fair bit of data. But I do think it is possible to do that now, because if we have multi-level data, and in the next few years, I think we're going to have other kinds of data. We're going to have omics data. We're going to have very large scale, and this is already happening to some degree, web-based assessments of very large populations, and you can get things that are not symptom counts, but also performance kinds of measures, things like Test My Brain on very large samples. Integrating those, I think, would allow us to fractionate, and it's going to probably take a variety of statistical and machine learning methods, but then you need to show from other criteria that it helps predict treatment or so. Again, a lot of work, but I think it is feasible to do in a way that you couldn't have done 10 years ago, because we just didn't have that variety of data. And then the last thing I'll say about that is, in the case where we have rare genetic variants like copy number variations or rare mutations, then I think you actually can get subtypes maybe that are more clearly biologically defined to some degree.
Jordan, thank you so much for this sort of gripping talk. I mean, I just think it was phenomenal.
Thank you.
Thanks for joining us this morning. And to celebrate your escape from Harvard, we're going to send you home with a UC San Diego School of Medicine psychiatry hat. Thanks again.